So, first of all, I'm really excited to be here. Most of my academic trajectory has been about integrating across fields. So the field of complex trait genetics involves a number of things, not just plant and animal breeding, but also population genetics, genomics, statistics, evolution, human biology. And so I'm really excited about seeing Inbreeding because this basically quantifies something I'm very interested in, which is talking across fields, and also something I'm quite interested in, especially which is education, as we still work on the. So an example, as we're waiting for the PDFs to display here, as an example of interdisciplinary interactions and how they're important, several people have mentioned the importance of animal welfare and welfare in general. And in fact, there's very nice tie-ins between animal welfare on one hand and a very different field, the evolution of altruism in behavioral ecology and evolution, and then a big debate about kin versus group selection. And all those nicely tied together in a general framework. And you can see that if you read the different literatures and put them together. So for example, we do that in chapter 22 in the book. So what I want to do though today is basically talk about some new sort of tools from population genetics and genomics and how these can be important to breeders. So basically I want to talk about using information from natural populations to improve plant and animal breeding. So here's the outline. So I want to briefly remind you about local adaptation of land races and lines. I want to talk about how breeders can exploit that variation. And I specifically want to focus on how we can use molecular markers to detect genomic regions under selection. I want to look at the patterns they generate and then how we can detect those. And then I want to talk about what some of the implications that are for domestication, something called sweeps. And I want to conclude with about using gene editing informed by natural variation. So my goal here is basically to introduce you to a pretty technical area in kind of a relatively, gentle overview fashion. And if you want to really dig down the details, chapters 8 and 9 of our book cover those. And as Sonar pointed out, I brought a copy. I'll just leave it up here in the second row. I'm not worried about someone walking off with it because they'll throw their back out by the time they get downstairs. So breeders want to exploit natural variation. It's why we talk a lot about the idea of preserving genomic diversity. And wild relatives, relatively undomesticated lines, and land races potentially contain vast treasure troves of minimal genetic variation. And this variation comes in kind of two flavors. Flavor number one is variation in production traits that genetic mutants, for example, that aren't present in current elite lines, but which are hiding in some of these land races. Those are certainly interesting, but even more interesting is the idea you can basically have variation in local land races that's a product of local adaptation. And if you can use that local adaptation, it might be to specific challenges, environmental features, that current elite lines are struggling with. So I'm going to mainly focus on this second source of variation, adaptive variation in localized lines, and ask how can we detect that, and how can we then move that into the current elite lines we have, both plant, animal, tree, shrimp, whatever you want to talk about. So to kind of set the framework, on what timescales can a line locally adapt to its environment? And the answer is it can do this pretty quickly. So here's the basic breeder's equation, kind of rewritten. This is response in terms of phenotypic standard deviation. I is the selection intensity, and then h squared, of course, is heritability. We kind of summarize a lot of literature from natural populations, and an average value of selection on a trait under selection corresponds to basically culling about the lower 5%, so saving the upper 95%. That gives you an intensity of about 11%. If you take a typical heritability, it means that one generation of response to natural selection changes the mean by about 0.3 standard deviations. So a 3 standard deviation change takes about 100 generations. So that's short on the time scale of most land races. So land races can very quickly, even under relatively weak selection, adapt to their current environment. So this raises the question, number one, how can we detect such variation? And more importantly, how can we exploit that as a breeder? And the answer is actually very classical. I think Sewell Wright was the first to propose this, but if someone else has an earlier reference, I'm very happy to hear about that. And this is the simple select and backcross. And the idea is very straightforward. You can do this before you even knew about DNA. You basically have a wild line of interest, and you have an elite line, and you have some environmental target that the elite line is having trouble coping with. Water stress, temperature stress, disease resistance, and you cross the local and the elite line, and then in the F1 you simply then select for individuals which have some resistance to your environmental target. Then you take those individuals and you back cross to the elite line, and you continue this cycle several generations. And what basically happens is, because of back crossing, you're removing almost all of the wild background except those regions which are under selection, and you're then introgressing most of the elite lines. So this is a procedure we could use not knowing anything about genetics other than, the transmission works. And the problem is, can we improve on this? And the answer is we can. And one way we can improve on this is to use marker information. And we can break marker information into kind of two categories. So again, when I talk about the trait being selected, I'm talking about an environmental target that the local line is adapted to. You're trying to introgress into the elite line. So if I can identify a trait, for example, specific disease resistance, or maybe salt tolerance, or high aluminum soils, or handling extreme stress heat stress if you're an animal, if I can identify a trait, or maybe it's an index of traits, I can then use standard QTL or association mapping to detect markers associated with that trait. And then I can use those markers when I do repetitive backcrossing. A second approach is what I call trait-independent approach, and this will be the focus here. Suppose you're not really sure what the target trait giving the adaptation is. You want to, for example, have a certain line of chickens being now grown in a new environment. You want to introgress elite production traits, but you want them adapted to an environment the elite lines have a hard time in. It's kind of unclear what the actual targets of selection there are, what the actual traits are. So trait-independent approaches use genomic information to look for markers which are involved in local adaptations. So I'll be talking a lot about this approach. And this comes out of population genetic theory, where there's a large literature basically on how to detect these things, mainly coming from the human side, but it also has recently been used in studies for searching for domestication genes. So suppose we go to the first approach, which is I specify an adaptive trait. Water stress, heat stress. And so then I can either select on that trait in my backcross scheme, or I can try to find markers, and therefore I can score those markers and skip the entire phenotypic trait, basically doing genomic selection. So the idea is I could use a QTL study or a GWAS, find those markers, and simply put those into a marker-assisted selection. And I could be extra fancy. I could, for example, delimit a region of interest, and then use speed methods to use outside markers so I can introgress things in faster. That's the basic idea. So if I do that with the standard QTL approach, so cross two lines and look for chromosome segments which are marked, what I typically see is large signals of large effect. The problem is those are usually bad signals. They're inflated by something called Beavis effects. Bill Beavis showed when you condition on an effect being significant, you tend to overestimate it, and you can overestimate it quite dramatically when the power is low. Likewise, when you take these large signals and you try to fine map them, a single large bump fractionates into two smaller signals. If you take one of those smaller signals, they continue to fractionate. So that one large signal is probably caused by a linkage block of a large number of different genes together, and as you select on that block, that one signal, that linkage block decays away. So QTL mapping have large confidence intervals on the order of 20 to 40 megabases or more. So those are big chunks. If I'm trying to introgress in an elite line, I want a small region of uncertainty about my adaptive markers, not a large region. So the bottom line is you could use a QTL study to find the adaptive trait and use marker-assisted selection. The problem is this has all the same features that old traditional marker-assisted selection have. You typically don't capture enough variation, and you basically have too large segments that you're introgressing. Well, maybe we can improve this with GWAS. Genome-wide association studies takes our region of uncertainty from megabases to kilobases. So they're much smaller regions. They require not a cross, but basically a population survey. That's an advantage. But the problem is the GWAS effects tend to be smaller. Now this is misleading because when you get a small GWAS effect, what GWAS measures are how much variation is associated with the marker. And you can have a large effect allele that's very rare, and as a consequence of that, you'll have a small variance. So a small variance doesn't mean large effects alleles aren't out there, it simply means they're rare. So you could again use a GWAS. Again, I specify a trait, do a GWAS, and then in my select phase of select and backcross, I select on those markers for the adaptive trait. Now the problem with both of these trait-based approaches is that you've got to specify the specific trait. In some cases that may be very obvious. In other cases, if I'm trying to adapt to a new environment for the elite line, that trait or traits, we either may not know what it is or we may choose the wrong target or only partially pick the target. So is there a way to get around that? And one way is use trait-independent methods, which use genomic scans. So look in my wild line and see what chromosomal regions show evidence of relatively recent selection, and those would suggest targets to adaptation to its environment, and we can basically then use those. So this leads to the question basically of how do we detect those? And as I mentioned, there's a ton of theory on this which gets a little bit complicated. I want to kind of give you an overview. And the idea is that when you have recent selection, it generates a pattern in genomic data. That pattern could be something as simple as reduction in the amount of diversity, or it could be more subtle in reconfiguring the way diversity is found. And a number of tests look at those features, and they basically rely on this idea called a coalescent. So the coalescent is kind of the idea that modern molecular population genetics is built around. It's an idea by John Kingman in the 80s, and it's basically this. So what this figure here shows is I've got 1, 2, 3, 4, 5 DNA sequences that I sample right now. So I've got 5 sequences at a specific gene. If I go back in time, those 5 sequences, well, these 2 alleles have a common ancestor back here, so I've gone from 5 sequences down to 4 ancestors, and as I go further back in time, they coalesce, and eventually they coalesce back so that all the 5 sequences derive from some distant ancestor. And the nice thing about these, if these are neutral alleles, there's a very specific, nice mathematical theory about what this pattern looks like. The reason it's important to us for detecting selection is here's just an allele down here. So here are 2 alleles I sample. If there's a long time back to a common ancestor, these red ticks are mutations, and what you see is that long time back to a common ancestor results in these 2 sequencing differ by 5 mutations. If I have the same mutation rate but a shorter time back to a common ancestor, these 2 sequences differ by a single mutation and therefore show less divergence. So one of the things that selection does is it shrinks that time back to the recent common ancestor and therefore changes the patterns you see in polymorphism. So the idea for this is something called hitchhiking. I have a chromosomal region on which a new mutation which is adaptive that arises. Then that adaptive mutation is driven by selection to high frequency, ideally fixation, and as a consequence, it drags along nearby sites. Plant breeders call this linkage drag. It's the same idea. We also talk about hitchhiking. So a neutral allele linked to a selective site can hitchhike up to high frequencies. This results in all those sites having a relatively recent common ancestor. Recent common ancestor, not a lot of variation. It sweeps out the variation in a window around that selective site. And we call this a sweep. A partial sweep is the signal that happens when that allele isn't driven to fixation, but kind of goes a little bit of the way there. It generates a pattern, not as dramatic as this. And finally, we'll talk about a polygenic sweep later on, which is the pattern that generates the kind of weakest signal. So basically, here's the idea. Here is a normal coalescence structure for a neutral allele. Here's what it looks like with the selective sweep. So if you imagine it, there's a lot more variation between these sequences down here than over here. Why? These all trace back to a very recent common ancestor. This is a case where that site has swept to fixation in the population. If I look over here, a partial sweep, I see this structure looks quite different from that. And there are two things that happen with selection. Number one is you change the total length of that. So you make those coalescences shorter. Making them shorter means you have less variation. That's kind of the obvious part. The more subtle part is you also change the topology. If I shrink this down to be that size, this thing looks nothing like that. The topology also changes. And so not only do you change the amount of variation, but you also change features in how that variation is patterned. And a number of tests, so-called site frequency tests, are basically built on that. So let's take these rather abstract notions and give a concrete example. One of the classic domestication genes is TB1, teosinte branched 1 in maize. Here's the ancestor of modern maize, teosinte. It's a multiple branched species or population. Modern maize has a single branch. There is a gene in modern maize called TB1 that gives it a multiple branched pattern. So it's a candidate for a domestication gene. Now, if you look around that gene, the cartoon we would expect is I go across a chromosome, and around the selected sites I see a dip or a sweep in the background variation. So I have a little window here, and this lack of diversity in here is a signal that that region has been swept through, it's been under selection. So what does a region look like in TB1? Well, here, the darker line here is variation in teosinte. The dashed line is variation in maize, and what you see in the teosinte branched 1 gene itself, the patterns are pretty similar, with maize being a little bit less. You expect maize being a little bit less because maize is a subset from teosinte, it's gone through a bottleneck. The interesting feature is let's look upstream. Here's the variation you see in teosinte. There's no variation in maize. So it's basically, this is another way of looking at this pattern. So there's a sweep, but teosinte branched 1 isn't the target. The target turns out to be a hopscotch retrotransposon that's 64 kb upstream. So this selection here gives us this pattern of reduced variation upstream of teosinte branched 1. A clear indication that selection has occurred there. Another example, dogs, Dachshunds. These little short limbs, there's a gene in humans that causes limb shortening, and here it is. If you sweep across it in dogs, what you basically see is in control groups, there's also a dip in variation, but there's a dramatic dip in Dachshunds. So obviously, the gene that was swept through to give short limbs in here affects this region. By the way, one thing you'll notice is this region is asymmetric. It's not symmetrical. So you can't simply take a region of reduced variation and take the middle point of that as the reduced spot of variation. So these are examples of how we can use some of these basic methods to detect domestication genes. But the same approach can be used in natural populations. So how do we use these in a statistical sense? You can think about that amount of reduced variation being a race between two forces. Number one, selection. Number two, recombination. So allele frequency changes as kind of 1 over S. So if S is large, allele frequencies change very quickly. If S is small, allele frequencies change slower. So the time scale for selection is 1 over S. The time scale for recombination is C. So you'd expect the effect of a sweep to be some ratio of C over S. And in fact, you can actually, this is a general parameter that tells us at a specific position away from that sweep, what's the reduction we see, and you see it's a function to basically C over S. So that's the foundation for a number of tests we'll talk about in a second. So you can take results from here basically and ask the first question, which was, what's the range of a sweep? There's a nice result here due to Dick Hudson and Norm Kaplan. It gives you the length of the sweep as a function of the recombination rate average over that region and the strength of selection. So for example, if you take a typical figure of 1% recombination per megabase, so 1 centimorgan per megabase, that's a pretty average figure. What you see is if I have an allele that has a 5% selective advantage, the sweep covers 100 kb. If it has a 0.5% of advantage, the sweep covers 10 kb. So what you see is this trade-off between recombination and strength of selection. The larger the effect, the larger the window. So we can also turn that around and ask retrospectively, if I find a sweep region, what's been the strength on that region? When you do that for Teosinte branch 1, you see it's about 5%. So the selection that was on that maze architecture was about 5% of the, that just gave you about a 5% advantage, pretty small. Now you can take this result here and ask what happens when you have inbreeding. So a lot of important land races in plants are inbred, and inbreeding reduces the frequency of heterozygotes, therefore it reduces the effective frequency of recombination. And the net result, if you just take a window down here, let's say a 5% selective advantage, with 50% selfing you go from 100 kb to 150, but with 99% selfing you now are in the megabase range. So basically, if you have inbreeding, it makes that signal much, much larger, which is good. It makes it easier to detect, but it's bad news because you can't really localize it. So there's a trade-off between the larger the region you have, the easier you can detect it, but large regions are not useful for integration because they contain other stuff besides the target. So you can have a number of tests based upon maximum likelihood that basically give you where that target region is going to be. If you actually work them out, what you see is the resolution is on the order of about a half to a quarter of the total sweep width. So if you go back here, the resolution here is on the order between 25 and 50 kb. So the upshot is the resolution of these sites under local adaptation, the resolution is about the same as a GWAS. The advantage is you don't have to specify a trait in advance. So if you have a localized line, you can scan that localized line, find regions of selection. You won't find them all, but find some regions of selection. And the level of resolution there is about on the order of a GWAS, but you're not making any assumptions about what the actual target traits are. So that's an advantage. So that's one category of tests. Basically, I ask, how much reduction and variation do I have? I'll briefly mention another because if you read the literature, there's a lot of discussion on these. And this is a concept called the site frequency spectrum. Suppose I have a DNA sequence, and in that DNA sequence I've got, let's say, 200 sites that show variation. I can then look at site number 1. Suppose I've got 1,000 DNA sequences, and I can ask, what's the number of the minor alleles in here? So maybe it's 50 out of 1,000 are minor alleles. The next one is 1 out of 1,000. We call that a singleton. The next one is 400 out of 1,000. And so I can go through each site and basically construct a histogram or a frequency spectrum of how many are present as just 1 copy, 2 copies, 3 copies, 4 copies. And there's an expression called the Watterson Distribution that specifies those. And the bottom line is it's given as a function of 4 times the population size times the mutation rate. Now, when I have selection, that frequency spectrum is changed. So this dotted line here is the Watterson distribution. When I have selection, it basically forces it this way and that way. The solid line is this distribution following a sweep. And you see, a little hard to see, but there's an excess of rare sites. There's also an excess of high frequency so-called derived sites. So the site frequency spectrum gets changed by selection. And a lot of tests use this. And those tests basically follow along these lines. This Watterson distribution depends upon this parameter theta, and it turns out there's a lot of ways to estimate that from data. One way is I could simply look at my entire sequence and count up how many of those sites are segregating. Well, that S, I can write that then as an estimate of theta, where I have theta times something that depends upon the sample size. I can take 2 DNA sequences and I can ask about the average number of pairwide differences between them. That also gives me an estimate of theta. Or I can simply ask what fraction of all those sites have a singleton, that is, a rare allele is present as only 1 copy. All those 3 things are different aspects of that site frequency spectrum. They should all give the same estimate of theta if that site frequency spectrum is neutral. If it's not neutral, we'll give different estimates. So by comparing these or several other different estimates with each other, and here are their variances, basically t-test type things, there's a number of tests. I won't bore you with these except to mention that one is Tajima's d-test, and the other more populars are Fei and Wu's H and Zhang's e-test. So these are all tests that use genomic data and they used a fine structure of that variation, not simply a lack of diversity, to ask, can I get a signal of selection? So that's another category of test that you can use to try to detect either local adaptation or, if you want to, either domestication or improvement genes. So as I mentioned, this area is rather complicated because you stack layers and layers and layers upon it. Looking for depressions and variations and changes in the site frequency spectrum, all that theory assumes something called the hard sweep. And a hard sweep says you're sitting around and all of a sudden a mutation arises as a single copy. It's adaptive. It sweeps through the population. So here's that single copy, and it drags along that same haplotype to all the other individuals when it's fixed. Another possibility is something called the soft sweep. A soft sweep says that mutation arose, but initially it's either neutral or maybe even slightly deleterious, and it bounces around the population for a while. Then the environment changes. All of a sudden that mutation is now favored. And in that case, a soft sweep, what you see is that instead of a single type being fixed, you basically fix a population that's a collection of all these individual haplotypes. So as you might imagine, the signal from here and the signal from there are quite different. So soft sweeps give you different type of signals than a hard sweep does. And the other way to think about hard sweeps versus soft sweeps say we have adaptation from existing variation. Hard sweep says we have to wait around for an adaptive mutation for the population to change. So there's a lot of interest in hard versus soft among evolutionary biologists. So how do we then detect these soft sweeps or a partial sweep where the allele doesn't go to fixation, it goes partially there? And the key is linkage to disequilibrium. And here's that idea. So here is my starting haplotype. I've got a SNP there, and around that I've got a block that's in linkage to disequilibrium. Now, over time, as that allele increases in frequency under drift, it's going to be older, and therefore recombination is going to make that haplotype smaller. Under neutrality, a common allele is an old allele. An old allele means it's been around a while, and therefore there should be shrinkage in the amount of linkage to disequilibrium around it. However, if I see a common allele with a long haplotype, those are not compatible with each other for a neutral allele. A common allele means they're old, a long haplotype means they're new. So there's a large category of tests for detecting partial or soft sweeps, which are basically based upon not looking at variation, either site frequency spectrum or depletion of variation, but rather looking at the structure of haplotypes. And this just kind of gives you, the idea here, and again this is from the book, the idea is that there are a number of different signals you can use depending upon what your sweep is. And so what you're probably getting right now is that we would like there to be a single test, like a QTL or a GWAS. Unfortunately, because selection can occur at several different phases, different sites, one may be a hard sweep, one may be a soft sweep, one may sweep to fixation, one may not sweep to fixation. So the net result is no one single test works, and we have to have a collection or a cocktail of tests to do this, and somehow combine those. So when you do that, you again get resolution about on the order of about 50 kilobases. So these tests which can detect sites under selection, number one, don't detect all the sites, just like a GWAS doesn't detect most of the sites. But number two, their level of resolution is about on the order of a GWAS. Now, the worst scenario happens in this case where you don't have one or two genes that give you adaptation. You've got a large number of genes, a small effect, scattered across the genomes that each have small rises in frequency. That's called a polygenic suite, and that's the most difficult to detect because you have soft sweeps, partial allele frequency changes, and weak effects. And to give you some idea about that, this is if I have a QTL that has an effect on an allele, we can express that effect in terms of standard deviations. So this term is the standard deviation effect that specific allele has on the trait. There's the selection intensity. That's the selection experience that allele sees. So if I have a trait under modest selection, intensity of about 10%, an allele with one standard deviation gives me an S of about 0.1, so I should get about a 200 kb signal. But an allele with a tenth of a standard deviation now gives me a signal on the order of about 20 kb. So polygenic sweeps are notoriously difficult to detect because they have smaller signals. Recently, there's been a new approach from Jonathan Pritchard's lab called the Singleton density score. And the idea is to use whole genome sequencing. Everything I've told you before you can do with kind of chips. You don't need to have a whole genome sequence. With the whole genome sequence, I've got a region of interest. I've got, let's say, 100 sequences. I take a target region, and I take that target region, and I scan how far I have to go over on either side until I see a singleton. A singleton means a variation that's only found in one chromosome in our entire sample. And you can look at the ratio of those to the two alleles, and that gives you something called the Singleton density score. And what Jonathan showed is this is actually relatively efficient for detecting polygenic sweeps. His data was looking at English data from the English medical records, and the sample size of 3,000 individuals, they could detect ongoing events at about 2%. So the bottom line from all this is, number one, to introduce you to some of a huge variety of machinery that can be used to detect ongoing selection. So number two, to point out that there's different tests required for different phases. So no one single test does them all. The result is you have a cocktail of tests. In the event that you detect something, you have resolution that's somewhat poor on the order of 50 kb. So we're back in the situation of poor resolution and only a subset of possible regions. So we kind of have the same problem with marker-assisted selection. So from a breeding standpoint, we can use machinery to detect regions of interest. If they're major genes, we're fine. If they're minor genes or genes which haven't gone all the way to fixation, it's going to be hard to utilize them. So the net result from kind of digging deeply into population genetic literature is this method, which initially seems rather promising, is basically no better or no worse than a GWAS. But I don't want to leave you on a down note. So the obvious question is, are there other ways to use genomic data on adaptation? And the answer is there are, and they're actually quite interesting, and they get into gene editing, which I guess you can't do in Europe, but let's assume for now that you can do that, and we'll talk about that. So if I have a region, so I've got a chromosome, I've got a target on it that's selected during domestication or improvement, that target generates a sweep around it. That sweep around it will drag along other genes. It's called linkage drag. Bill Hill and Alan Robertson, in a paper that was initially fairly obscure, but now it's the paper that we use to analyze any genome, made an observation that other people, namely Joe Felsenstein, called the Hill-Robertson effect. And the Hill-Robertson effect says, if I have a site here that's under selection, a nearby site will have its selection be less efficient. So if it's deleterious, it'll be more neutral. If it's advantageous, it'll be more neutral. And the net result on that is selection at one site makes a nearby site less effective, so linkage drag can drag along deleterious alleles associated with your adaptive site. And there's examples that happen. A good example is rice. So if you look relative to wild rice, domesticated rice shows regions often over 200 kb of reduced diversity. Now, when I say reduced diversity, most rice lines are highly inbred. So what this means is I have a panel of lines. I look over that panel of lines. In that panel of lines, I see reduced diversity. A good example is the vaue gene. The vaue gene is a splice mutation that gives you sticky rice in some groups of Japonica. And that region or sweep of reduced diversity covers about 250 kb, or on the order of 40 genes. So selection by breeders has dragged along potentially deleterious mutations. And the best example of that comes from rice. So a group compared Japonica and Indica with the wild ancestor in an outgroup. And when you look at the wild ancestor versus the outgroup, and look at either Japonica or Indica versus the outgroup, you found a higher rate of replacement, that is amino acid altering substitutions to silent substitutions, than you see for Rufipogon. So domestication has increased the rate of amino acid substitutions, changes. Now, you might think, oh, that's just adaptive genes being driven along. Most adaptations are probably regulatory. There are examples which are clearly amino acid changes, but a lot of them aren't. So what this suggests is a potential signal for domestication dragging along deleterious alleles. And what these authors then did is they looked at these in Japonica and Indica and showed that a disproportionate number of them involved radical substitutions. So 2 amino acids which aren't biochemically similar, but rather different. That's a classic red flag that one of those may be deleterious, and they estimated that about a quarter of the replacement substitutions in domesticated lines were likely deleterious. This is the so-called domestication mode. So as a plant breeder or an animal breeder, you're making your elite lines at some level potentially a little bit sicker because you're dragging along all these deleterious recessives with them. Well, this is where gene editing and natural populations can come in. So in theory, if I could target a deleterious allele in an elite line, I could edit it and improve the elite line without additional breeding. Well, the obvious question is, how do you find those deleterious alleles? The answer is, this is where natural populations come in very cleanly. I can have a large sample of variation in a target region from a natural population. Then I can look at my elite line that's fixed for that, and I can ask, is that substitution in the elite line, is that found in natural lines? If it is, I'm not going to worry about it. If it's not, or it's extremely rare, it's an obvious candidate for editing. And so those obvious candidates for editing, then, you can be basically informed from the natural populations. So natural populations potentially provide a lot of information about specific targets to edit, because in natural populations, deleterious alleles will be rare. Now, obviously, this won't be 100% accurate, but it's a good initial first step. And all it requires you to do is basically take a wild population sample and sequence from that. So in summary, methods to detect ongoing and just completed selection exist. They allow you to find trait-independent markers for adaptation. The level of resolution is finer than QTL and roughly on the order of a GWAS. So using detected markers really only works with major genes. Now the flip side for natural variation is sweeps from domestication improvement can be quite impactful. This is the domestication load dragging deleterious linked alleles to fixation. We can use genomic data from natural populations to edit those. So one interesting way to talk about regulators in the future about gene editing comes into the context of welfare. We think about animal welfare, well, there's also plant welfare. As a breeder, you may have made your elite lines sicker than they should be by dragging along these deleterious alleles. You can be a physician to the gene pool and replace those deleterious alleles with their copies found in the natural populations, and conceivably in some cases, significantly improve the well-being of those lines, be they animal lines or plant lines. The summary final slide comes from my hobby as moth collecting, which Anar referred to, and this is how people like to think about the genome. What you want are these big, giant, wonderful things. That's your idea. If you find those giant major genes, marker methods work great, either by trait-based marker methods or by marker methods based upon simply scanning and looking for sites under selection. In reality, a lot of the variation you see is probably due to a whole bunch of things with much smaller effects. So breeding works really well from a marker standpoint. You have genes of really large effects. If you have genes of really small effects, you're in the genomic selection framework, which is essentially asking what fraction of chromosome segments do individuals share. So the bottom line is, what I would try to do is cover the intellectual exercise of what we can use modern population genetics for to detect adaptation in natural populations. And the bottom line was the answer is somewhat unsatisfactory. But that's good to know, so when you want to try it, you know what your limitations are. The other side is natural populations are extremely informative as to whether or not deleterious alleles have made it into our elite lines. And with that, I'm open to questions. Thank you very much. And I'm under time.
Yeah. So, thanks a lot for this wonderful talk, a lot of food for thinking, and we're just now open for questions.
I wouldn't believe everything I said.
maybe I can start with.
Sure.
So in a way, all the conceptual idea is based on the idea that adaptation is due to a single gene or to a group of single genes. I think there is some evidence that adaptation basically is an adaptation of gene networks and of complex things. What to do then?
Yes, that's a great question. This gets us to this notion about hard sweeps versus polygenic sweeps. And so you might imagine adaptation about being like this. So you may have a couple of genes of large effects involved in your adaptation. Those you can pick out. And you could have a whole bunch of, maybe far more in terms of biomass, of smaller effects genes that together have a much larger effect. That's the category of the polygenic sweep, where each of those could leave a signal, but that signal requires special things to look at, namely things like Singleton density scores. So when I frame these methods, I've talked first of all about detecting major genes, and then detecting genes whose effects get smaller and smaller and smaller. Just as with marker-assisted selection, the bigger the effect of your gene is, the easier it is to deal with. And unfortunately, most genes probably have relatively small effects.